name is Dominika Gruszka. I am a Sir Henry Dale Fellow, funded jointly by the Wellcome Trust and the Royal Society, and I am a junior group leader in biological physics here at the Kavli Institute for Nanoscience Discovery. So my research group seeks to understand how epigenetic information is maintained during DNA replication. So DNA is the carrier of genetic information. Each human cell, on average, has about 2 meters of DNA. That's the length of DNA. In order to pack that into the cell, it would be impossible to just wrap the DNA around itself. It's quite a long linear molecule, right? So chromatin is a solution to it. So you employ dedicated proteins called histones. And what histones do, they are essentially like a little spool around which the DNA is wrapped. And then these little balls, nucleosomes, they pack against one another in three-dimensional space and they form chromatin. And within that, how condensedly they are packed will depend on whether you want to access genetic information encoded within the DNA or not. So those loosely packed, you can read the information. Those that are more tightly packed, they are inaccessible to the cell. And that is essentially how different tissues read their genetic material. On a slightly more global level, chromatin is partitioned into distinct functional domains, and these domains control cells' access to genetic information encoded within its DNA. And that in turn controls cellular function and so-called cellular identity. Now, nucleosomes that are located in these different chromatin domains carry specific epigenetic labels, and these epigenetic labels control chromatin structure and chromatin domain dynamics. Now, DNA replication is a process by which the cell duplicates its genetic material prior to cell division. And during DNA replication, chromatin structure undergoes a complete structural rearrangement. And this is because in order for DNA replication to occur, the DNA double helix needs to be opened up and the two strands need to be physically separated. Nucleosomes that are located in the cell have to be disassembled in order for that process to occur. So from the perspective of chromatin, the process of DNA replication is a process of both yin and a yang. So ahead of the replication machinery, nucleosomes are disassembled from parental DNA. But then once DNA replication is complete and the two daughter DNA strands have been synthesized, then the exact same nucleosomal landscape and the associated chromatin structures need to be faithfully re-established on the daughter DNA in order to maintain cell function and cellular identity. Now, if something goes wrong during DNA replication, and for one reason or another these chromatin structures are not correctly re-established, then this can lead to onset of diseases such as cancer. So my work sits very much at the interface of biology, chemistry, and physics, and Kavli provides all the essential facilities for me. But what I cherish most about the Kavli is its collaborative and inclusive culture, and I have benefited from that already. So even though I have been here for only 6 months, I've already established some absolutely amazing and exciting collaboration across physics, chemistry, and biology.